Okay, so now we'll just go over some examples of multi-omics integration from from some cancer studies and then an L1000 study. So this first example is predicting breast cancer chemotherapy response. This is from a paper published in Nature last year where they were using multi-omics, namely genomics and transcriptomics, coupled with some pathology information to try and characterize the tumor ecosystem and then use those features to predict chemotherapy response for breast cancer patients. So they did this whole genome sequencing and transcriptomics profiling of 168 breast cancer patients, and the outcome they were looking at was a response to chemotherapy. So either complete response where no tumor was observed, or some level of residual disease. So the cancer came back basically. And they were measuring neoadjuvant chemotherapy. So this is chemotherapy that's administered before the surgical resection. And so the first thing they did was, because they're interested specifically in how the tumor ecosystem predicts chemotherapy response, this is sort of a dimensionality reduction slash, using prior biological knowledge sort of approach. But they took all of these omics data and kind of grouped them into features specific to breast cancer and what they're interested in studying. So tumor features would be, would estrogen receptor and HER2 expression, as well as the mutations present in the sample, and measures of chromosomal instability. And then these different immune processes were also characterized from the transcriptomics and genomics data through like gene set enrichment analyses. So these are leveraging known pathways or known immune-related pathways, and finding, how much of that are we observing in the transcriptomic data. So this is a way of sort of they were condensing all of that very rich transcriptomic and genomic information into these biologically meaningful features that are then used for prediction. So their initial exploration of those individual omics features, they found that individually they were not robust predictors of treatment response. And so this sort of prompted in the approach of integrating the features across different omics layers to try and build a better prediction model. And what they found was that the best performance was obtained from combining all of those omics features, so the DNA, RNA, in addition to those digital pathology features, and that this led to the best prediction of chemotherapy response. So I think on the right here is just the feature importance that was computed for each of these clinical and omics related features. But this is an example of how multi-omics integration can really provide stronger and more accurate results than a single omics analysis might. So the second example is coming from the LINKS program, where they're characterizing the response of this breast epithelial cell line, MCF10A, in response to the binding of these 6 different ligands. So these 6 ligands were chosen based on their high phenotypic impact. So the phenotypic outcome that's being measured is cell morphology, and which is characterized with this immunofluorescence staining. But then the other omics profiling that was done to try and understand the molecular patterns driving these phenotypic changes were RNA sequencing, which was done with, or gene expression, which was done with RNA sequencing and L1000 assays. We also did epigenomics characterization with ATAC-seq and as global chromatin profiling method. And then some proteomics were also included through this reverse phase protein array. So there's a wide range of omics that were captured. And the goal was to try and relate those omics profiles to the observed phenotypic change in this cell line in response to the binding of these ligands. So basically, a lot of those features from each omic were extracted through a differential expression analysis. So the cells were first observed at time point 0 before any of the ligand was introduced into the media, and then after the ligand was introduced, all these omics were captured at 5 different time points over the span of 48 hours. So we kind of get this temporal picture of how the cell is responding as a system to the ligand binding. And one of the, I think, important ligands that were studied here is this cytokine, interferon gamma, which is very important in a wide array of both innate immune and adaptive immune responses. And it's been a huge focus of study just for trying to understand the role of the immune system in disease pathophysiology. So through this differential expression analysis at each omic layer, we can perform enrichment analysis that is essentially serves as some dimensionality reduction technique. So we went from that large gene or protein expression space into this smaller feature space. And then we use protein-protein interactions to connect these different omics layers and put together kind of this network of signaling pathways that may be happening and driving this phenotypic response in these cells. So we can construct these different networks over different time periods. And this network was built by Alexandria Keenan from the Mayan lab. So we see here in the rectangles, we have these enriched transcription factors, which are inferred from the transcriptomics data. We have them connected through protein-protein interactions to those up and down proteins that are coming from proteomics, as well as these up and down transcripts coming from transcriptomics, and these other inferred molecules that sort of are mediating these interactions. So through multi-omics characterization, we now have kind of this integrated sort of multi-layer capture of all the different signaling that might be happening inside the cell in response to interferon gamma. Okay, so the third example is multi-omics target prediction through this multi-omics to target pipeline, which was recently developed by the Mayan lab, and it enables this integrated analysis of transcriptomics, phosphoproteomics, as well as proteomics to predict both cell surface protein targets and driver protein kinases that are then integrated for personalized therapy, or that can be targeted for personalized therapy. So the overall approach is a hierarchical integration approach. So we're leveraging existing knowledge about transcription factors and their targets and kinase substrate interactions to transform all of those multi-omics data into one common feature space that reflects kinase activity. And we do this using differential expression, clustering, gene set enrichment, and some network analysis methods. So the workflows that are involved in this pipeline are, we have 3 different omics that we're using: phospho, transcriptomics, and proteomics. So in the phosphoproteomics component of this approach, we take the up and down regulated phosphosites, and we use KIA3, which is an enrichment analysis tool for kinase substrate interactions to rank the kinases most likely responsible for those up and down phosphorylation or phosphosites. So we get this essentially a ranked list of kinases prioritized by how abundant their substrates are represented in our up and down data set. And then we kind of compare that to what we see from transcriptomics. So this is sort of where a hierarchical approach to data integration or to multi-omics comes in. So we start out with transcriptomics, where we have these up and down regulated gene signatures. And then using what we know about gene expression regulation, we perform transcription factor enrichment analysis with KIA3, which uses libraries of transcription factors and their targets to rank the transcription factors that are most likely responsible for those observed changes in the genes. So now we've connected genes to transcription factors using these libraries, which are primarily from ChIP-seq experiments. And then using protein-protein interactions or co-expression, so these are where other omics can start to be introduced. We can expand that network of transcription factors with literature-based biomolecules that we know are also observed with those transcription factors. And then that expanded subnetwork is what we then use for kinase enrichment analysis, similar to what I described earlier in the phosphoproteomics pipeline. But what that gets us is kind of from the transcriptomics space, we've now through like biological knowledge and this hierarchical approach, we've brought it into this kinase enrichment space. So we have a measure of kinase activity for each sample. Okay, so that's the kinase prediction multi-omics workflow. And then the cell surface target prediction is this separate component that takes transcriptomic data. We first perform some clustering of that transcriptomic data into subtypes. And then we do differential expression analysis. So we compare the expression in those clusters to these background data sets of normal, healthy gene expression across many different tissues and cell types. And the goal is we want to find those genes that are highly expressed only in tumor cells and lowly expressed across the rest of the tissues in the body. And this is ideal for if we want to design some kind of targeted therapy that selectively binds only to those tumor cells without interfering in any other tissues. So once we've identified those genes, we want the protein products of these genes to be localized at the cell membrane. So since these are accessible by therapeutic, so like CAR T cells or antibody drug conjugates. And we do that through applying some membrane filter, which relies on proteomics-based localization of those gene products to the cell membrane. And then if proteomics data are available, we can also incorporate that as kind of an additional filter. So we're choosing the differentially expressed genes that also have protein expression that is, or proteins that are abundant and localized at the membrane. So this is how we use both transcriptomics and proteomics, and knowing, I guess, how they confirm or validate each other to predict ideal gene targets for immunotherapy. And this workflow is implemented by another tool we've developed called Target Ranger. So a big part of building this Multiomics to Target pipeline was making it available as a website and making it user-friendly. So this is a lot of work from Giacomo, who built this Multiomics to Targets website. And what it enables is it accommodates a combination of any of the three omics. So this analysis will run if there's just transcriptomics or just proteomics, as well as any combination of 2 or 3. So that kind of enables maximal flexibility with depending on what data the user has available. And then additional features that are part of this site are independent validation of those gene and kinase targets. So we use CRISPR screening data from DepMap. So this is a cancer-specific validation, but we use that as sort of an independent measure of whether those predicted targets are shown to be like meaningful for the tumor cell survival. So this is kind of an extra measure of is targeting this gene or kinase going to be effective in removing the tumor cell. And then another component of this website is an output report that's presented as a fully complete research paper with abstract, introduction, results, discussion, etc., as well as those complete figures and text. And as with some of the other tools we've been building, we're using these large language models, so GPT-4 in this case, to compose some description of the results and try and make sense of what we observe in the data. Okay, so just to summarize some of the different topics and then integration approaches we've talked about today. So first, multi-omics integrate data from these various omics regulatory networks or regulatory layers, which includes genomics, transcriptomics, proteomics, metabolomics, and lipidomics. And by putting them all together, it provides a more comprehensive understanding of those biological cellular systems that we're observing. And when it comes to the challenges of multi-omics analysis, data integration is a big focus. We have a lot of difficulties that arise from data complexity, heterogeneity, as well as just challenges to the management and infrastructure of those data. And then due to the complex relationships that we've discussed between these different omics layers, machine learning approaches have now been very widely adopted for the integration of multi-omics data. And we've gone over some of those reference omics databases that are very valuable tools for reducing the dimensionality of those raw, like complex and very high dimensional omics into something that's more meaningful and smaller dimension. And then lastly, single cell multiomics and spatial multiomics are an emerging area of research with very promising applications moving forward for providing that full picture, comprehensive view of the translational and transcriptional, translational, and broader characterization, I guess, of what's happening inside a system. So this has really important implications just for biological research as a whole and translational medicine. So I put here some resources and references for all the information in this presentation. I bolded this last one here is I think a nice summary of just of all the different tools that are out there and how they're the different categories that they fall under. And then for specific coding related tutorials for multi-omics integration, that was a little more challenging to find. And I think most of the packages out there are in R. So I linked here a nice R tutorial. Okay, and then yeah, I want to thank Avi for the feedback and content on these slides, and then the rest of the lab for their support, and we'll gladly take any questions if there's any.